Hello, I'm Michael Majurski. I'm here on behalf of the NIST ISG team to present our image stitching tool, MIST. Brief outline for today's presentation. I'm going to discuss the 2D stitching problem. Is this a little bit? Okay. Then we're going to cover the novel aspects of MIST, translation optimization, how to validate a stitching tool's accuracy, and our computational acceleration results. Finally, I'm going to cover some stitching examples. I've got a pre-recorded demo video. I'm not brave enough to try to do it live. And then I've got the tool availability links, and then any questions. Brief overview, the 2D stitching problem. Take a grid of partially overlapping image tiles and convert them into a single output mosaic to then do further processing on segmentation, feature extraction, something more interesting than just the math. First part, the novelty of MIST. The MIST algorithm is 3 basic steps. You have the translation computation, translation optimization, and mosaic assembly. The translation optimization is where MIST has been focused our work on. The first step computes translations between every image in the grid and its adjacent neighbors. MIST uses a phase correlation method taken from the image stitching literature. Next, the translations are optimized using a model of the mechanical microscope stage to compute the stage backlash and repeatability. These values are then used to limit the translations, bounding the error in the stitched image. The image is then finally constructed using a minimum spanning tree by putting together the optimized translations. And this again is taken from literature. So the novel part of MIST is just in how the translations are optimized. Novelty 2, how to validate stitching accuracy. No single tool will work for all data sets. Therefore, we created a validation methodology outlining how to collect ground truth data and use it to evaluate a given stitching tool in order to determine which performs best for your test data. Stitching methods are evaluated using distance error, size error, false positive and false negative counts. The distance error is done by computing ground truth distance between intercolony distances to the stitched intercolony distances. For MIST, we acquired 3 test data sets of stem cell colonies over varying image overlaps and sizes. And the ground truth data was acquired to get the positions on the stage and the sizes of the colonies within a single field of view. On average, MIST is a distance error of less than 2% of a field of view and a size error of 5%. The size error, false positive, and false negative rates were primarily due to photo bleaching as the samples were acquired. The novelty 3, computational acceleration. As the world moves towards big data, more and more processing takes longer and longer, and as things move towards the cloud, you got to optimize for speed in some cases. The image stitching problem is massively parallel. It was a great candidate for stitching for acceleration. The MIST implementation as an ImageJ plugin uses all available compute resources. By default, it uses native FFTW libraries. And if your user has a compute-capable NVIDIA GPU, you can enable its selection as well. To highlight MIST runtimes, I've got 8 datasets shown here with GPU, CPU, and the Fiji grid collection stitching runtimes. The computer used to generate these had a mid-range CPU, mid-range GPU, and the images are stored on a hard disk, not an SSD. Okay. The data set number 4 in the table, the IPS cells, is a representative example of the stitching performance between the GPU and the CPU. On average, you get between 1.5 and 2x when you use the GPU. And for most stitching experiments, we found that it takes longer to set up the GUI parameters than it does to actually run the stitching, until you get to the really big data. A few stitching examples. MIST has been applied on diverse data sets acquired across 5 different microscopes, generating over 1,000 stitched images with varying grid sizes and overlaps. The majority of the image content so far we've seen has been cells in cellular biology, but it's not limited to that. We're expanding into the material sciences, and we've got some examples up here of paper with embedded microparticles, SEM carbon nanotube images, and some live worms. The biggest data set we've seen so far is a 70 by 93 image tile grid, which turns out to be 93,000 pixels on a side. Through time-lapse data, we got a big experiment with 3 replicas of 18 by 22 image tiles in the grid at 161 time points and 2 channels, each replica about 350 gigabytes. And that one took probably an hour and a half to stitch. I've got a pre-recorded demo. Let's see if I can get this to work. Launching MIST. That's kind of, okay, that's kind of weird. Basically, standard plugin launch, and it opens up the GUI here. This input tab is broken up into 3 pieces. The acquisition setup is how you've organized the images as they were acquired in the microscope. The input folder controls where the files are stored on the disk, and multi-channel stitching below that is assembling new images with old translations. For the test data set, you've got two types of grids, sequential and row column. Micromanager has the row column format. Then you've got to set up your grid origin, upper left, lower right, lower left, lower right. The graphic updates as you go along. For this test data set, the grid width is 10 and 10, and if you're doing a row column, you can press discover and it will automatically look at the disk and figure out what's there. Next, you want to set up the image file name, and we've kind of matched the Fiji stitching plugin for file formats using the curly brackets to denote iterators. And I've got the file paths selected here and saved off in a text file so I don't have to mistype them. Next, you're going to check your output settings. Where are you going to save the output image? Generally, I like to stuff it in a subfolder of where the input tiles are to keep everything contained. You can rename the output prefix. It gets appended to every file MIST will generate. You can adjust the overlay blending between the images, and you can display the image or you can save the image, and those toggle both. Check the subgrid because you can stitch just subgrids of the image instead of doing the full thing. You can do a test 5x5 to see if you've got the parameters right. And then you want to check the advanced tabs. I'm here just making sure there's nothing extra there. The default values only. And then we're ready to go. And so we go down there and click Begin Stitching. Now MIST found a few files it was going to overwrite, so queried for confirmation before overwriting them. You click Yes, and the stitching will launch. And this is 100 images being reassembled into a single composite. And it's blending them, building the real image in Fiji, and it'll display it in a second. Okay? So like I said, it took longer to set up the grid parameters than to actually stitch the image. This is my backup in case the demo didn't work. Now the MIST is available as a Fiji update site, so you all know how to manage that. Just go to the update sites, click Check MIST. The source code is freely available on GitHub. We've also got a pretty extensive wiki, and you can visit our project page at isg.nist.gov. Thank you. Do you have any questions? Yes? Not currently. That is the next thing we have to tackle. This is simply a 2D stitcher. You can do it through time or multiple channels, but you can't do like a 3D volume yet. Sorry, what? No, that is, again, we've got that in MATLAB, but we don't have that in the ImageJ plugin. Like the one example we had in MATLAB was a circle inscribed in a square, and we can handle that with the MATLAB code, but we can't with the full Fiji plugin. Again, it's on the to-do list. Any other questions? Yes? That's the whole point of the optimization is you determine from the translations between images what the mechanical repeatability and backlash are. And if you have higher magnification, those values will be relatively larger in the pixels count. So the higher the magnification, those values will be bigger and there's more error, but it should still be able to work it out.
Yeah, but then if you have like vibrations from the environment,
Yeah, again, the optimizer it searches a box. So once the backlash and the repeatability have been estimated, it searches that box to find the optimal translation. And the hill climbing optimization of the normalized cross correlation should find at least a local maximum, if not the global, for that translation pair. Yes?
Your GUI seems more complex than the normal stitching in Fiji. So is it still scriptable?
It's still scriptable, yes. You can use the recorder to determine the parameters to rerun this thing. We kind of didn't use the generic dialog for Fiji. We went a different direction to get a little more complex in the GUI because we just like the look of it. But you can still script it. It's hooked into the macro language. Yes?
Two quick questions.
Yeah.
Data structure for all of this internally, I see you use IJ processor for some stuff. Is that what you do, or is there something else?
Once you actually get into the Java code, it's all internal. There's no the data structures are being managed as a memory pool of image tiles, and the FFT data is tied to the image data. And so that's the only thing we're using from ImageJ is the actual TIFF reader or the file reader.
Okay, so this is multi-place.
It's mostly TIFFs, but I mean PNGs. Anything that the ImageJ reader can handle, this can handle. We've used it on 32-bit floating point images. I've used it on PNGs before.
How hard do you think it would be to update to the more general data structure internally?
Internal?
It would really give you a lot more.
It would take actually a complete redesign, because the speed is gained by managing memory very carefully and streaming it through. So we have to do a certain optimization in how the gradient image tiles are read and the processing chain. So it would take a redesign to update it to the n-dimensional data structure. This was more towards the speed side, and we had a specific problem case. We didn't go for the general case.
So if you have TIFFs, use best.
Yeah, TIFFs, PNGs, any small two-dimensional image, we're good here. If you got a complex 3D volumes, we can't handle that yet, at least. Anything else? Thank you.